topic that we are going to deal about is about the history of microbiology. That is the historical development of microbiology. So before going on with that, let us know what is microbiology. Microbiology is the study of microorganisms that are too small to be seen with the unaided eye. Human eye has a capacity to perceive object of diameter 1 mm. Less than 1 mm, human eye cannot perceive it. So which type of organisms are usually included under microbiology? So it is the organism which have 1 mm diameter or less than 1 mm diameter are included under the broad domain of microbiology. The examples being the viruses, the bacteria, the fungi, algae and the protozoa. So algae and fungi, only the lower groups of algae and fungi can be included under the broad domain of microbiology. So it was only after the invention of microscope that we are able to know about the invisible group of microorganisms. So it is a microscope that provides us with a magnification by combining different lenses that enables the specimen to be observed in an enlarged form so that the human eye can perceive it. So it is only after the invention of microscope by different scientists that we are able to know about the broad domain of microorganism. So the foundation for the microbiology comes from the thinking that the diseases are caused by invisible group of organisms. So people did not have a knowledge about the group of organisms that is which are minute in nature that cannot be seen with the unaided eye. But they have a thinking that these diseases are caused by certain type of invisible creatures. So that laid foundation to the group of microbiology. So it was in the 13th century that Roger Bacon said that diseases are usually caused by invisible creatures. So before that it was thought to be due to the supernatural powers or wrath of God that usually the disease is caused in human beings. But it was in the 13th century that Roger Bacon proposed that diseases are caused by different groups of microorganisms which are invisible creatures. So in 1546 it was Fracastoro of Verona who said that contagium vivum is the one, the invisible creature that is contagious in nature and causes the disease. That is, if a healthy person are usually placed or the sick person, if it is placed in a group of healthy people, the healthy people also will get sick. So that is contagious in nature. So he said that it is contagium vivum that usually contagious and it causes diseases in the human being. And in 1762 it was von Plensis, he said that different diseases are caused by different agents. So we can see that slowly the concept of the supernatural theory was being overlooked by the organisms that are usually involved in causation of the disease. That means people had a sense of understanding that there are certain creatures that are able to cause disease but not due to the supernatural powers or wrath of God. So these type of development help to know about the invisible creatures that are usually present in the environment that causes the disease. And it was Kircher who said that minute worms which are invisible in nature can cause are the responsible for the different types of diseases. So it was only after 1665 where Robert Hooke was able to see the cork cell under his microscope. He said about the minute things that are less than 1 mm in diameter. That is, he was able to study the cells under his microscope. And it was in 17th century when Antony van Leeuwenhoek, who was a drape merchant in Delft of Holland, who had the hobby of grinding lenses and producing magnification and studied different natural things under his microscope. So during his study, he had seen the rainwater drop under his microscope containing minute organisms that were moving in the raindrop. So he called them animalcules, and hence we attribute or we give the tag to this Antony van Leeuwenhoek as the discoverer of the microbial world. So here we can see the presence of the organism from the 13th century to ultimately discovery of the invisible world with the development of microscope that was done by Antony van Leeuwenhoek. So it was Antony van Leeuwenhoek, who is called as the discoverer of the microbial world. So during his lifetime, he was able to produce 200 to 300 microscope of different magnification, and this helped to identify organisms. So along with the development of microscope, he was able to try to see different types of organism under his microscope. The development of microbiology took a setback after his death due to technical difficulties, and it was resumed again in 1884 when a compound microscope was developed, and hence the study of microbiology also began after 1884. And now let us see what are the historical developments to see how these organisms have been included under the broad domain of microbiology. So the first thing that had to be cleared off was the spontaneous generation versus biogenesis theory. So there are two classes of thoughts over here. There are two different scientists that usually believed in spontaneous generation theory or the biogenesis theory. According to the spontaneous generation theory, the life usually exists without a living cell. That is, no pre-existing cell is required for the life to come up, or the life usually develops spontaneously from a decayed products or non-living things. So this was a school of thought that completely believed in spontaneous generation. That is, they thought that there is no requirement of pre-existing cell for the life to generate. So even the great Aristotle belonged to this class of thought of spontaneous generation theory. So later this was refuted or challenged by a scientist called as Francisco Redi, who usually observed different, he was a naturalist who observed different things around the nature. So he found that maggots usually develop from the eggs, and they ultimately develop into the flies, and it is the egg that acts as a pre-existing cell to develop into the fly. So he carried out an experiment in which he placed 3 jars, and into which he placed a piece of meat, and one was left uncovered, the other was covered with a paper, and the third one was covered with fine gauze of cloth. So ultimately he could see that the growth of the organism in a jar without any cover, the flies was attracted to the meat, it laid the eggs, and finally maggots were developed. Whereas in the jar with the paper, there were no maggots developed, and then third jar he could see that the flies could not have an access to the meat. So they laid the eggs on the gauze, and from which the maggots were developed. So he concluded his experiment saying that life will not exist spontaneously but will come from a pre-existing cell. That is, in case of flies, it is the eggs that are laid, and which gets converted into maggots and then ultimately to the fly. So the first to refute the spontaneous generation theory was Francisco Redi. So people then began to think about the spontaneous generation theory was like it's absurd, and they began to take up with the biogenesis theory. And like with the development of microscope by Leeuwenhoek and the discovery of the microorganism, the concept of spontaneous generation was shifted from plants and animals to now bacteria. So now people of spontaneous generation theory have believed that bacteria develop spontaneously from the organic matter. That is, in earlier days, bacteria were usually grown in a meat broth or the infusion from the meat broth. So it was first John Needham who has usually carried out an experiment in a jar which was tightly corked, and then he boiled the broth and left it for some time and observed at regular intervals. So you could see that initially the broth was clear, and later on it developed different organism and make the solution turbid. So when he observed this under the microscope, he could see it teeming with the microorganism. Ultimately, he said that while boiling eliminates all type of organism, all type of existing cell. It is that broth which is the organic matter that produces the bacteria, and it has a vegetative force or a vital force to develop bacteria. So there is no pre-existing cell that is required for the bacteria to develop. So it is directly coming from the organic matter of the meat. So this was challenged by Lazzaro Spallanzani, who doubted the method of heating procedure that was used by John Needham. So he carried out experiment in which he has boiled the broth with a tightly clogged jar and for continuous 1 hour, and he repeated the same experiment, and finally he could see that no organisms developed in that particular jar. So this was again challenged by John Needham, saying that while long time boiling we could destroy the vegetative force that is present in the organic matter, and ultimately you are eliminating the vegetative force, thereby no organism can develop. So in order to contradict John Needham, he has broken over the seal and he continued boiling the broth, and finally he could not see any growth of microorganism. So at the same time in the chemistry departments the development was towards the oxygen. So oxygen was known to be the vital part for the survival of any cell. So this was taken as a plus point by the spontaneous generation people to say that while boiling for continuous period of time, you are eliminating the oxygen that is providing the vegetative force to the organic matter to develop the bacteria. So later on there were two scientists who was called as Theodor Schwann and Franz Schulz. They passed the air through different materials. So they carried out the experiment to prove that air can provide the growth of the organism, and it is the air that usually carries the germs that has a pre-existing cell to develop the bacteria. So in Theodor Schwann, what they did was they took the two jars containing the meat broth, and through one jar they passed the air with a red hot tube, and the other jar was taken as a sample which was where a direct air was allowed to pass. So but obviously they could see that it is only the uncovered jar and the jar in which the air was passed directly developed the organism, whereas the jar in which the air was passed through the red hot tube did not contain any microorganism. So they said that air has been provided but there is no growth of organism. So similar type of experiment was done by Franz Schulz, where in a beaker of the meat infusion the air was passed through the chemical, that is concentrated sulfuric acid and sodium hydroxide. Here also they could not see the growth of the microorganism. But again the spontaneous generation people had challenged, saying that the air has been manipulated or morphed, due to which the vital force is not getting the vegetative force to develop the microorganism. So later on it was Schroeder and T. von Dusch that has introduced the cotton plugs, that is passed the air through the cotton plugs. Now the abilities of the cotton is that it provides pure air to let in, taking away all the germs on its other end. So only the pure air could enter into the jar, and hence they could see that no organisms were developed inside this particular jar where the meat infusion was fed. So some people started believing biogenesis theory, which states that air contains the organism that usually helps to produce organism in a meat infusion. So after certain years, it was in 1859 where the spontaneous generation theory was revived again by a scientist called as Felix Pauchet. So he has carried out experiment where he provided the air without any change, and he could see the growth of the organism. So ultimately the final blow to the spontaneous generation theory was given by Pasteur, who was then working with the fermentation processes. So he was the one who was able to identify organisms which are involved in the fermentation process. So he had the concept that these microorganisms are present in the air and dust particles, and when they land up on the organic matter, they are able to produce into cells or the colonies. So it was Pasteur who gave a final blow to the spontaneous generation theory, where he has prepared a swan shaped neck flask, and through this the air was passed, and due to the gravity of the earth, the dust particles settled in the U-shaped tube, and only the pure air could lead into the flask, and thereby not allowing the growth of the organism. And when this flask S-shaped swan neck was broken, you could see the growth of the organism. So that was a final blow that microorganisms will not develop spontaneously, but they will develop from the pre-existing cell. And these microorganisms are present in nature as ubiquitous, and they are present in every forms and every genera and species, and when they land upon an organic matter, they usually develop into, or they reproduce to form colonies. So with this, blow was given to the spontaneous generation theory, and the use of organism in different field was, the significance of organism in the different field is now known to us. So let us see about the importance of organism in disease causation, or which is nothing but the development of medical microbiology. So germ theory of diseases. So initially, as discussed, most of the diseases are usually caused by the germs. But before, people used to have a belief that it is due to the supernatural powers or the wrath of the god that usually causes the disease. So this was classified as supernatural theory. Later on, people began thinking that either due to natural causes or due to the pollutions in the environment that can lead to the causation of different diseases. So it was in 1546 when Fracastorius wrote a book called as De Contagion, where he published about the organism which is nothing but Contagium vivum. That is, they are able to cause contagious disease, that minute organism that can cause diseases. And it was in 1835 where Agostino Bassi, who has proposed that muscardine diseases in silkworm is caused by fungus. And in 1843, it was Oliver Wendell Holmes and Semmelweis who were able to prevent childbed fever, which is called as puerperal sepsis. So they said that this childbed fever is usually contagious in nature, which is transmitted to the woman by the doctors or the midwives. So the best way to avoid this infection is to wash hands with the disinfectant. So people began thinking about the organism and the use of organism and causation of the diseases. So it was Louis Pasteur who finally entered into this field and studied about the different organism that causes the disease. Previously, he was working with the fermentation process and was able to identify organisms which are used in fermentation to produce different fermentative products. And he was the one who had solved the problem of souring of wine. So not only France but whole Europe was looking up to him to solve other areas of microbiology, mostly the diseases that are caused by microorganisms. So he was able to identify and isolate organism that causes pebrine disease, anthrax disease, chicken cholera and hydrophobia. That is nothing but the rabies diseases, which is caused by dog bite. So these are the organisms that are causing the different diseases. So he has carried out different demonstration in public to show that organisms are the one that causes the diseases. And during his studies, he also told about the vaccines and the attenuated strains of organism that can be used as vaccines. So in order to honor Edward Jenner, who laid a foundation to these vaccines, Louis Pasteur gave the term vaccines to the attenuated strains that can be used to produce immunity in an individual. So vaccines are nothing but antigenic preparations that are inactivated and given to raise the immune status against a particular antigen or a bacteria. So it was Louis Pasteur who was very well identifying organism of different diseases. Similarly, as Louis Pasteur was carrying out experiment in the France, at the same time it was Robert Koch who was identifying bacterial techniques or bacteriological techniques to identify bacteria, prepared culture media, and has given us a concept of pure culture to identify organisms in the lab. So it was Robert Koch from the Germany who had given us a concept of bacteriological techniques, pure culture, staining techniques, and he was also the one to identify the organism that causes anthrax disease, that is a bacilli, and also a bacillus of tuberculosis, which is causing the disease tuberculosis in human being, and then chicken cholera. So during his studies, he proposed certain postulates that helps in identifying the organism in the lab, which is called as Koch postulates. And it was his student Henle who had given a concept of Koch postulates, and it was ultimately proposed by coach to identify the organism in the lab. And later on, many developments were carried out in medical microbiology, and there were students who belonged to Louis Pasteur and students belonging to Robert Koch who identified different organisms in their respective labs. So in 1874, it was Hansen who identified leprosy bacilli. 1879, Neisser who identified gonococcus. 1881, Ongston staphylococcus. 1884, Loeffler discovered diphtheria bacilli. And in 1884, again, it is Nicolaire who identified the tetanus bacilli. And in 1886, Frankel pneumococcus. 1887, Bruce Malta fever. And 1905, it was Shodin and Hoffman who was able to identify the organism that causes the sexually transmitted and a very lethal disease that is called as that is caused by spirochete, and the disease is called as syphilis. And till here it is only the organism that causes the diseases, but later on when the organism was studied in detail, they found that it is not only the organism that causes disease, but in certain products produced by the organism that can also have pathogenic effects. So these are called as toxins. Toxins are nothing but metabolite products of the organism that have pathogenic characteristic. So certain organism produces toxins, and this toxin when reached to a particular target might inactivate that target. So they are the organism that produces the toxins are diphtheria. So diphtheria produces a toxin that can act on the human body and cause central nervous disorders. So it was in 1888 Ross and Ursin isolated the toxin that is produced by diphtheria bacilli, and ultimately it was Ehrlich who gave a biological standardization of toxin and antitoxin. Antitoxins are nothing but the neutralizing antibodies that are produced against the toxins that are produced by the microorganism. So when an antigen enters, the human body reacts by producing the antibody. Similarly, when an antitoxin is usually entering, it is the when the toxin is entering, it is the antitoxin that usually will go and neutralize the toxin. So it is about the microorganism, that is bacteria till here. But there were certain infections that were not known, such as influenza, rabies, etc. So these are usually caused by filterable agents. Pasteur, though, has isolated the organism, like recognized the organism that is causing rabies, but was not able to isolate in the lab. So he said that there are certain creatures that are minute than bacteria that are able to cause the infection. So it was ultimately Ivanovsky who was able to demonstrate the filterable agents in the TMV, and that is tobacco mosaic disease, and the organism that is causing the tobacco mosaic disease is a TMV virus or the tobacco mosaic virus. So ultimately it is not only the bacteria, but there are certain components that are minute than bacteria that are able to cause the infection. So later on development in the virology was taking place, and the foundation was laid by Ivanovsky. Later on Bajering coined the term as viruses, and in 1898 Loeffler and Frosch first isolated the organism foot and mouth diseases of cattle, and the first human disease caused by viruses is an yellow fever caused in the people of Cuba. So in 1902 it was Walter Reed who was able to propose us about the yellow fever caused by the viruses, and in 1909 it was Landsteiner and Popper who isolated the organism poliomyelitis, and it was only isolation till now, but after the discovery of microscope, electronic microscope, that we are able to study the characters of these viruses. So it was in 1934 when Knoll and Ruska discovered the electron microscope, the detailed studies of viruses were carried out. So apart from that, the cultivation of viruses in the living cultures, that is the chick embryo. Now this the basic difference between bacteria and viruses is that viruses cannot grow in an artificial media. They require a living host because they are very minute organism consisting of only two components, protein and nucleic acid. So they require a living host, and hence they are called as obligate intracellular parasite. To grow this organism, we require a living cell. So the first culture technique that was carried out to isolate viruses in the lab was given by Good Pasteur, where virus cultivation was done in chick embryo. From then, there were different viruses that were usually cultivated, and various vaccines were produced against this particular viruses. And in 1917, it was Trott and D. Herrell who identified the bacteriophages. So it was only viruses till now. Now we know that viruses not only cause diseases to the human beings, but they also have the ability to attack the bacteria. So viruses which attack the bacteria are called as bacteriophages. And this concept of bacteriophages is very important in typing the bacteria, because not every virus goes and binds to the bacteria. There are certain receptors on certain bacteria where the viruses can go and bind and cause the infection. So it was not only the human beings but also the bacteria that can be lysed with the bacteriophages, and also the plants have different types of viral receptor. Therefore, the virus which attacks the plants cannot attack the human beings, and the virus which attacks the bacteria cannot attack the plants. So they require certain receptors. So it was Trott and D. Herrell who identified the bacteriophages. So this is about the development of medical microbiology, where we know about the different organism and the causative agent of different diseases. Now coming to immunology, next broad domain of medical microbiology is immunology. Immunology is a study of immune system, where different components of immune system, such as organs of the immune system, cells of the immune system, and the antigens, antibodies, and antigen-antibody interaction that helps to eliminate the antigen from the body are studied. And the basic concept of immunology was given to us by Lady Mary Wortley Montagu in 1718, where she proposed a concept of variolation to protect against the dreadful disease which is called as smallpox. So she added a small amount of smallpox ingredients from the pus to the skin, and this small amount of smallpox provided immunity against the dreadful disease which is a smallpox. So that is variolation, which is a bit lethal when compared to that of the other concept that was proposed by Edward Jenner. He used immunization to smallpox by cowpox. So he had seen that the person who was working with the cows, they were affected with the cowpox, which is less lethal to that of the smallpox. So the dried material from the cowpox pustule was introduced into the boy who was suffering from the smallpox, and this provided immunity to that particular boy, and that concept of immunization started. So helps to eradicate the dreadful disease which is a smallpox. And it was Pasteur who was able to identify and prepare the vaccine against the diseases of various infections. So in order to honor Edward Jenner, he had term it as vaccines. Vaka means cow. So because it was Edward Jenner who had given a concept of the mechanism behind how the immunity can be raised by introduction of the cowpox against the smallpox. So it was Pasteur who had produced different types of vaccines. And in 1888, it was Nuttall and who said that defibrinated blood has bactericidal activity. So the concept of immunology was coming to know that there is an immune system in the body that helps us to protect against different types of infection. So 1889, it was Buchner who proposed that the serum contains certain types of factors which are called as alexins. Now we know that they are antibodies. And in 1890, von Behring and Kitasato has given us about the antibodies which are usually fighting against the antigens. And in 1895, it was Baudet who proposed us with the concept of complement system. So apart from antigen-antibody interaction, there are certain serum proteins which are called as complement proteins, and they completely belong to humoral immunity. So immunity can be broadly divided into two types, humoral and the cell-mediated immunity. Humoral immunity is B-cell mediated immunity, whereas cell-mediated immunity is T-cell mediated immunity. So we will be studying in detail when we study about the cells of the immune system. So it was Baudet who has given us a concept of complement system and proposed about the humoral immunity, and in 1883 it was Mechnikov who understood about the phagocytosis, that is the engulfment of pathogenic microorganisms by certain macrophages and elimination of the antigen. So he proposed us with the concept of cellular immunity by phagocytosis. And in 1902, it was Portier and Richet is said about the hypersensitivity. So as we know that immune system is the one that protect against different types of infection. So apart from that, the immune system sometimes can go awry and it can overreact to produce allergic response. So that is nothing but the hypersensitivity reaction. So there were different concepts that were proposed about the immune system. So one thing is about the hypersensitivity is it is an allergic reaction or exaggerated immune response where the immune system overreacts to a particular antigen. And in 1959 it was Thomas and 1967 it was Bernard who proposed us with immunological surveillance. Immunological surveillance is nothing but surveillance of different types of cells of our immune system, and there is a regulation or control of the production of this cells in our body. When this regulation breaks down, it can lead to a disastrous or deleterious effect in our body where our own immune cells will destroy our own body. So there is a regulation of this particular system, that is immunological system in our body, and that is usually done by immunological surveillance. And when this breaks down, then there is a deleterious effect that can ultimately lead to the death of that particular person. And Medavar and Burnett has successfully carried out transplants. Like once we know about the immunological status of a person and compatibility, then we can go for a successful transplantation. And this is usually done by not allowing the clones of T cells and B cells that acts against the tissue graft transplant or anything that is being transplanted. So this was successfully carried out by only after the discovery of blood groups by Landsteiner in 1900. So he was able to classify blood groups into 4 different types, and the compatibility transfusion was also taken up with this particular logic that was proposed by Landsteiner. So this is about the immunology, which is again a branch of microbiology. Now coming to the other branch, which is called as fermentation, that is the industrial microbiology. Now these microorganisms are so ubiquitous in nature that they affect human life in all areas. So whether it is soil, whether it is disease causation, or whether in producing important types of materials or metabolites for human body, these are ubiquitous and they are helpful, and as well as they are more deleterious to the human beings. So here coming to the fermentation, the development of industrial microbiology. Fermentation is a biological process that is carried out by most anaerobic group of microorganisms where metabolites are produced by these organisms that are important for the human beings, such as alcohol, we have acetic acid, we have citric acid, etc., that are produced by the microorganism and used by the human purpose. So it was Theodor Schwann in 1837 who said that there was a role of yeast in alcoholic fermentation. So yeast was responsible to carry out fermentation from the sugar to produce the alcohol, and it was supported by Lauter and Cushing, and later Louis Pasteur entered into the field, and he had solved the problem of soaring of wine in the French industries. When he studied the fermentation processes, he could identify that organisms are able to produce alcohol which causes the soaring of wine. And when studied in detail, he could see that there are different types of fermentation caused by different types of bacteria. And with the same concept, he came up with the pasteurization, where heating of the milk to an extent to eliminate the microorganism without destroying the quality of the milk. So this was initially used for pasteurizing the wine. Nowadays we used in the dairy industries. So apart from this fermentations or industrial microbiology, the organism also has contributed majorly to the soil cycles, biogeochemical cycles. So most of the organism, apart from causing diseases, causing the spoilage of the food, or being beneficial to the human, they also carry out different biogeochemical cycles in the nature. So the father of soil microbiology is Vinogradsky, who could propose different cycles that are usually carried out by microorganism in the environment. For example, nitrogen cycle, you have sulfur cycle, phosphorus cycle, where they usually convert different materials in the environment to be used by the plants, thereby increasing the soil fertility. So this is all about the historical development of microbiology. Now coming to the scopes of this microbiology, as these microorganisms are both harmful and beneficial, and they are present everywhere, they usually affect different aspects of human life. So study of organisms in disease causation helps us to identify the organism and treat different types of diseases, and to produce vaccine, the most beneficial effect of this organism. And for food preservations, you can make use of certain organism for synthesizing different types of products from the organism. So there are variety of fields by which the humans are usually have more importance of microorganism.